Now, the second part of this chapter that we're going to be looking at is something known as protein synthesis. In the previous videos, we have introduced DNA. We have also talked about RNA, the three types of RNA, mRNA, tRNAs, and also rRNAs. Now, what do these nucleic acids have to do? What are their functions? How are they important for our lives? The answer is all the nucleic acids, DNA and RNA, are important for one process inside the cell known as protein synthesis. And what exactly is protein synthesis? Well, to put it bluntly, it is just the production of proteins within the cells. Now, cells synthesize different types of proteins to carry out specific functions. For example, B lymphocytes will produce certain proteins known as antibodies to protect our bodies from infections. Goblet cells in our intestinal lining and also our tracheal lining will produce mucus, which is a particular protein that is needed to trap dust and pathogens in our airway or also our intestines. Erythroblasts, don't have to memorize this one though. Erythroblasts, which are the cells before they become red blood cells, erythroblasts will have to produce proteins known as hemoglobins needed to carry oxygens when they become mature red blood cells. And last but not least, beta cells in your pancreas, they have to synthesize proteins known as insulin, which helps to control our blood glucose concentration. So these are specific cells in our body, and they produce specific types of proteins that carry out specific functions. So the antibodies, mucus, hemoglobin, and insulin are all proteins, but they all have different structures and they all have different functions. As we've discussed this a little bit in chapter 2 when we talked about amino acids and proteins. So long story short, there are so many different types of proteins, right? So how does the cell know which protein it has to produce? Because a cell can have the capability of producing any types of proteins that I've just listed out, and then some. But how does the cell know that, oh, I need to produce this protein and not that protein? So before we talk about that in detail, we kind of want to cover a little bit about just an overview on the relationship between DNA, RNAs, and amino acids. If you want to study a little bit more on amino acids, you can go back to my playlist and check back chapter 2 where I talk about amino acids and proteins. Now, so for example, in eukaryotic cells or in eukaryotes where they have a nucleus, inside the nucleus, the DNA or the chromatin is found. And the DNA, as we've discussed, is just basically a type of nucleic acid that has a double helix shape. Now, specific parts of the DNA where I'm highlighting over there will be referred to as something called genes. So what exactly is a gene? A gene is basically a base sequence of the DNA that codes for polypeptides or that codes for a specific polypeptide. So a lot of times when I ask my students what's the meaning of a gene, students will say, oh, gene is a part of the DNA and it produces polypeptides. No, that is wrong. That definition is wrong. It's technically wrong because the gene itself does not produce the protein. The gene tells the cells how to build a polypeptide. That is a very important thing to know. It is more like a manual, okay? Like if you've ever assembled furniture or even built Legos, okay? When you're building Legos, like when you're putting the blocks together, when you look at the manual on which piece to put second and which piece to put third, the manual is acting as the gene. The manual itself does not build the Lego blocks for you. It just acts as a guide. That is what the gene is doing. Okay, it provides the information for the cell so that the cell can produce or synthesize the specific polypeptide or protein. Now, if the gene is the manual, then which part of the cell is supposed to produce the polypeptide then? The gene will have to provide its information to the ribosome because the function of the ribosome is to synthesize polypeptides. So For example, if the gene says, hey, make the polypeptide, but the gene is inside the nucleus and the ribosome is in the cytoplasm, they're so far away from each other, the ribosome cannot hear them. The ribosome cannot hear the order. So, because they are separated by the nuclear membrane, right? So, how does the gene provide the information to the ribosome? It provides the information to the ribosome by first copying itself or transcribing itself to produce something called the mRNA. How this happens, we will look at it in the next video. But long story short, you can just basically say the gene is transcribed or copied to produce something known as an mRNA. Now, what does the mRNA do? The mRNA will then move towards the ribosome. So, remember earlier where the gene and ribosome were far away and they cannot meet each other? The mRNA acts as the middle person. So, the mRNA takes the information from the gene and gives it to the ribosome. And once the ribosome receives the mRNA, the ribosome is like, oh, I now know how to make the polypeptide. But then comes the second problem. The second problem is, well, I know how to make the polypeptide, but I don't have the ingredients to make the polypeptide. So what are the ingredients needed to build the polypeptides? The ingredients it needs to build the polypeptides are amino acids. So because polypeptides are basically made by joining one amino acid to another. So where do the amino acids come from? Well, very simple. The amino acids will then come from tRNAs. And as you can see, I'm drawing out the tRNA over there. And the tRNAs, I'm just drawing out 6 tRNAs, okay? And each of the tRNA are attached to different types of amino acids, as I have represented by different colors, all right? So what the tRNAs will then do is, the tRNAs will then bring the amino acids to the ribosome, and as they bring the amino acids to the ribosome will then join those amino acids together to synthesize the polypeptides. So long story short, the gene is transcribed to produce the mRNA. The mRNA will move towards the ribosome in the cytoplasm. The tRNAs will bring the amino acids to the ribosome, and the ribosome will then synthesize the polypeptides that is needed by the cell. That's how it works. So that kind of answers the question on the relationship between DNA, mRNA, tRNA, and rRNA, which make up the ribosomes. So they all work together so that the cell is able to synthesize a specific polypeptide. Now, I've answered the question as an overview of how the cell produces a polypeptide. But how does the cell produce different types of polypeptides? The answer is quite simple. As you can see in this nucleus here, I'm drawing out 2 chromatins, okay, as you can see in the nucleus, and each of the chromatin has a different gene. The chromatin on the top has gene A, which I've highlighted in yellow, and the chromatin at the bottom has gene B, which I've highlighted in pink. I think that's pink. Yeah, that's pink. That looks like pink. Purple, pink. Yeah, sometimes I have difficulty with colors, but you get my point. Now, the difference between gene A and gene B is their base sequences are slightly different. Okay, the base sequences of gene A and the base sequences of gene B are slightly different. Let's not go into the details yet. So let's say, as you can see, I have tRNA molecules over here. Now, my tRNAs look a little bit different. I've just simplified the tRNA molecules. I like drawing them. I know they're supposed to have a clover shape. I get it, okay? But I just like drawing them in that square, okay? The circles of different colors at the top represent the amino acids, and the 3 bases at the bottom represent the anticodons. Do not worry if you don't understand my diagram. Just understand that I like to represent or symbolize tRNAs in this way. All right. If you still don't understand, don't worry. We will be looking at the process in detail in future videos. All right. But now just imagine them to be tRNA molecules. So what happens is there's a ribosome there. The ribosome is made up of rRNAs and some proteins. So First, let's say the cell wants to produce a particular type of protein. Gene A undergoes transcription, or gene A is copied to produce the mRNA. The mRNA will then move towards the ribosome, giving the information to the ribosome. Once the ribosome has the information, specific tRNA molecules will then move towards the ribosome, bringing its amino acids together. And when the ribosome receives the amino acids, the ribosome will synthesize the polypeptide. In this example, I'm going to call it polypeptide A. So gene A provides information for the cell to produce polypeptide A. Let's look at gene B. Gene B undergoes the same process. It will produce the mRNA by being copied or transcription. The mRNA will then move towards the ribosomes, and in this case, the tRNA brings the information to the ribosomes, and the ribosome synthesizes the polypeptide. But as you can see over here, when it produces polypeptide B, compare it to polypeptide A. Are they exactly the same? No, they are not. Polypeptide A is longer, made up of 6 amino acids, and polypeptide B is shorter, made up of 4 amino acids. And the amino acid sequences are different, as I've represented in the colors as well. So I'm sorry if you're colorblind and if you can't notice the difference between the color, but notice that the difference between the, what I'm just trying to say is the amino acid sequence in polypeptide A and the amino acid sequence in polypeptide B are different, because the information came from 2 different genes. So gene A will tell the cell to produce polypeptide A, which is a type of protein, and gene B will tell the cell to produce a different type of polypeptide, which is polypeptide B. That's how the cell is able to produce different types of proteins, because the cell has different types of genes. That's basically it.